Hello, we are a senior design team from Drexel University. Our project involves a groundbreaking technique of an automated method of mapping neural connections found in brain tissue. There are several large organizations dedicated to researching the brain, one of which is an NIH-funded project called the Brain Initiative, which has a budget of over $300 million. The goal of this project is to map the brain, understanding the structure and function through neural connections. One method of this is through connectome mapping, and it is used in disease research as well as other applications. Neurons allow for information transfer within the brain. Each neuron contains a cell body, which is where the cell nucleus is located, as well as axons and dendrites, which is how information flows and is received by other neurons. Through these structures, information can be transmitted and neural networks are formed. Networks of neurons can be imaged using histology methods and scanning electron microscopy, which creates a 3D image stack. From these images, a connectome map is created, where the nodes represent cell bodies and edges represent axons and dendrites. This is an image representation of the neural connections which exist in three-dimensional space. Current methods utilize software tools, but still require a human to manually identify neural structures and to trace along axons and dendrites. This process is so timely that the only organism to be fully mapped is a nematode worm, which is a very simple organism. In order to improve the timeliness of connectome mapping, and in addition reduce human error and bias, there is a need for an automated method which maps neural networks. With this identified problem, our overall objective was to improve upon the method of studying the brain by automating the connectome mapping of SEM images. The program we've written is an automated process which takes the SEM images, identifying the critical neural elements using geometric filters, and removes unnecessary structures while also preserving cell body and axon and dendrite information. It then stores this as a binary matrix, which serves as a precursor to a connectome map. Here's an image with all the identified structures overlaid onto the original SEM image. As you can see from the lack of white space, the program identified the majority of the elements present in the image. These are the qualitative results verifying the accuracy of our program. Because the dataset provided on the Open Connectome website did not provide annotations, we were unable to verify these results quantitatively. Instead, quantitative analysis was performed with simulated data, as shown here, which contains simulations of the critical neural elements we wanted to identify. From these quantitative results, you can see that our true positives were above 96% in terms of correct pixel identification, and errors were kept under 4%. Here's an image of our generated connectome map on the left, compared with the connectome map shown earlier. Though these maps do not represent the same dataset, you can see that our generated results represent the connectome map data similarly. Though our software is groundbreaking, we would in the future like to have access to annotated SEM data to quantify our results. Despite this downfall, we believe we have successfully shown that the automated process for creating connectome mapping is possible and should be considered as a viable alternative to current manual methods.